Hi anatomy students, this is Dr. Allis. In this short video, we're going to compare the two processes that your cells use to build proteins, the processes of transcription and translation. Before we can compare these processes to one another, we need to review what happens in each. When we talk about the central dogma, that's the technical name for how cells use your genetic information stored as DNA or deoxyribonucleic acid and turn that information into mRNA, which is messenger RNA. This process of turning DNA into mRNA is referred to as transcription. And the goal of transcription is to get a copy of the directions for the particular protein your cell needs and to get that copy out of the nucleus where the DNA lives. Once that copy has left the nucleus in the form of mRNA, it goes out to the cytosol to where we find ribosomes to assist with the process of translation. In the process of translation, we go from an mRNA sequence into a polypeptide, or a line of amino acids that are attached to each other with peptide bonds, forming a protein. Now that we remember what transcription and translation are, let's work together to fill in a table comparing these two processes. When we talk about the process of transcription, which is the first half of the central dogma, remember that transcription starts with the molecule called DNA. At the end of transcription, we've turned that DNA into an RNA molecule. The goal of transcription is to get a message out of the nucleus. The RNA molecule that I made in the nucleus is a particular kind of RNA referred to as mRNA, or messenger RNA. And that RNA was built by an enzyme called RNA polymerase. RNA polymerase built this mRNA molecule using things called nucleotides. Remember, when we're talking about building RNA, RNA does not have the nucleic acid thymine. Instead of thymine, RNA has uracil, U. So anytime you're building an mRNA strand, make sure that you're including uracil in place of thymine. Okay, so we've built RNA and we've sent it out of the nucleus. Now it's time for the second half of the central dogma, translation. In the process of translation, we start with a molecule of mRNA. That mRNA goes to the cytosol where it bumps into those small protein building organelles called ribosomes. The ribosomes will read that mRNA message using tRNA molecules. tRNA molecules can read what mRNA says, and tRNA molecules bring with them the amino acids that help us to interpret what the message says. At the end of translation, we've built proteins to assist the cell with doing its day-to-day function.